For several years now, our ecosystem has been threatened by a new form of pollution related to human activity: pharmaceutical discharge. Poured into wastewater, these contaminants pass through the meshes of the sewage treatment plants and are released into the rivers. They're likely to affect our environment by compromising the natural balance of the environment and the water use. But what are the toxic effects of such chemical exposure on the aquatic environment? In the framework of a new European Interreg program, the DIADeM project aims to develop an integrated approach to diagnosing the water quality of the Meuse River. The project consists of several work packages. Among them, the University of Namur is especially involved in the study of the effects of a mixture of pharmaceuticals frequently used by consumers on a biological model, the rainbow trout. The study is managed by a consortium of several French and Walloon partners. The experiment is carried out under laboratory conditions where all the physico-chemical and biological parameters are strictly controlled and where only the concentration of pharmaceutical substances varies. Prior to the troutlets' arrival for the experiment at the University of Namur, the aquariums are installed, cleaned and disinfected. The filters are then sown with bacteria which will transform ammonia, present in fish waste, into less toxic nitrogenized compounds such as nitrates. The experimental system consists of a semi-closed structure where the water is renewed by a low flow rate of clean water to evacuate nitrogenized waste. Now that the installations are functional, all that's left is to pick up the troutlets. They come from the Pierrot fish farm in Rogny in the north of France. The 600 fish are transported to the University of Namur in a 500-litre oxygenated basin. Upon their arrival, all the troutlets are doing well. They are distributed as soon as possible and with great care among the aquariums at a rate of 35 troutlets per aquarium. An acclimatization period of 10 days preceded the experiment so that the stress caused by the transport and the change of habitat did not influence the experiment results. The physico-chemical parameters of the water, such as the temperature, dissolved oxygen, the pH, ammonia, nitrites and nitrates, are monitored daily to ensure the animal's welfare and to limit residual stress. At the end of this acclimatization period, exposure to the pharmaceutical mixture may begin. The presence and the relative abundance of pharmaceutical compounds in the effluents of sewage treatment plants located along the Meuse were recorded by the Walloon Water Authority, the SWDE. So that the laboratory conditions are as close as possible to those encountered in the natural environment, the fish are subjected for 6 weeks to a chronic contamination based on a mixture of molecules. Based on the information recorded by the SWDE, the DIADeM consortium defined a mixture of 5 molecules: an analgesic, paracetamol; 2 anti-inflammatory drugs, diclofenac and naproxen; an anti-hypertension drug, irbesartan; and a neuralgic, anti-epileptic and mood regulator, carbamazepine. The stock solution mixture containing the 5 drugs is diluted in water in order to prepare less concentrated working solutions, which are delivered in the aquariums via a peristaltic pump. This pump allows small volumes to be injected at a constant flow of 100 microliters per minute. This device is thus ideal to mimic natural conditions, namely a continuous pharmaceutical residue intake through the effluents of the sewage treatment plants. 4 aquariums are used as control basins and will not be exposed to the drugs. 4 others contain the 5 drugs with concentrations ranging between 25 and 100 nanograms per litre according to the molecule considered. This amount corresponds to the one measured near the sewage treatment plant discharges in the Meuse River. The last 8 aquariums contain the same drugs at a concentration 10 or 100 times higher than the environmental one. During the 6 weeks of treatment, the behavior of the troutlets is observed daily. No abnormal behavior and no case of mortality were reported during the experiment. The troutlets tolerate the treatment very well without deterioration of their general health or their appetite. In parallel, each week, the concentration of drugs present in the water is controlled. A water sample is taken in each aquarium. This sample is then sent to the Walloon Water Authority in order to check the stability of the drug concentrations in water. It's important to maintain fixed concentrations throughout the experiment. The substance concentrations are quantified in high-efficiency liquid chromatography coupled with tandem mass spectrometry, LC, MS, MS. This technique makes it possible to detect these molecules at extremely low concentration levels of about a nanogram per litre. By comparison, that would account for a 500 milligram pack of paracetamol in 200 Olympic pools. Each week, fish from the different aquariums are brought to the CER group's Malroy, which is also collaborating in the project. Certain drugs, due to their chemical configuration, tend to accumulate in tissue and are thus found at concentrations up to 10,000 times higher in fish compared to the concentrations found in the surrounding medium. This phenomenon is called bioconcentration. Expert in the field of biomedical research, the CER group is responsible for evaluating the bioaccumulation of the contaminants in trout. Besides the analyses mentioned earlier, the troutlet's blood and organs will be sampled after 1 day, 1 week, 3 weeks and 6 weeks of treatment. They will be contained in Eppendorfs, sterilized, numbered and associated with a color code. Their analysis will highlight the potential effects of the pharmaceutical molecules on the immune system, the nervous system, the detoxification systems and the reproductive system of the fish. Hello everyone, thank you for coming to this second sampling. We'll proceed in the same way as we did last time, starting by sampling the blood and then the organs. For these samplings, the fish are anesthetized according to a procedure validated by an Animal Experimentation Ethics Committee. The plasma, as well as the organs, are then preserved at -80 degrees Celsius for later analyses. The study at the immune system level starts with the analysis of the white blood cells isolated from shredded spleen. For the following experiment, the cells must not be frozen and studied directly after sampling. Thanks to his flow cytometer, Stéphane Batoul from the University of Reims is able to analyze each cell of the sample in a fast and statistically relevant way. Thus, the abundance of each immune cell's population and their antimicrobial activity is measured and appears in the form of a graph called a cytogram. Then the blood samples are centrifuged in order to separate the blood cells from the plasma. Plasma will be used to quantify the activity of various key humoral proteins of the innate immunity, such as the lysozyme and the complement system. In addition, various genes involved in immunity and the inflammatory process, which could be a potential target for the anti-inflammatory drugs to which the fish were exposed, are analyzed. To this end, genetic material is extracted from tissues and the activity of genes is measured by quantitative polymerization chain reaction, or PCR. This biotechnology makes it possible to analyze the expression level of genes transcribed in a cell or a tissue at a given time. Finally, in order to know whether the drugs have an effect on the defense capacities of the organism against a pathogen, the fish will be inoculated after anesthesia with the Aeromonas salmonicida bacterium, a bacterium usually met in aquaculture 2 months after the beginning of the treatment. On the nervous system level, the effect of the drugs is studied on the neurotransmitters, such as dopamine or serotonin. Neurotransmitters are chemical mediators released by neurons during the transmission of nerve impulse. Their abundance will be evaluated by chromatography in the muscles and the central nervous system or in the brain. As far as detoxification mechanisms are concerned, the activity of various enzymes involved in the detoxification of xenobiotics and in the antioxidants production will be measured in the liver. Lastly, the potential impact of the drugs on the reproductive system will be evaluated. The dosage of sex hormones, such as estrogen and testosterone, is done by radioimmunology, a method based on the competition for an antibody between radioactive hormones and the hormones naturally present in fish blood. Other biomarkers related to the reproductive system will be analyzed, such as vitellogenin, a key biomarker of exposure to endocrinal disturbers, such as the contraceptive pill. And the expression of various genes involved in the development of the reproductive system in fish. Although the concentrations in pharmaceutical substances are maintained stable in the laboratory for 2 months, the experiment revealed that these substances accumulated in only small amounts in the trout tissue. However, biological responses were observed even in the weakest doses, comparable to those measured in the Meuse. For example, significant effects were observed on the reproductive system, such as a stimulation of the expression of the genes intervening in the development of the genital organs and blood concentrations in certain sex hormones up to 5 times higher than in the control trout, indicating a possible endocrine disruptor effect of this pharmaceutical cocktail. The study also showed that exposed trout developed an inflammatory reaction to drugs. In the long term, this reaction could induce tissue damage and hence a deterioration in the overall health of the body. The validity of these biomarkers is now to be confirmed in the real world, in fish in river cages, where they will be subject to many other potentially toxic substances.